Out of the 200 million sperm that begin the journey, merely dozens remain when they reach the massive, curved horizon of the egg. We usually picture this as a race won by a single, solitary swimmer. But a human sperm doesn't actually have enough enzymes to break through the egg's outer defenses on its own. To get through, they must function collectively. As shown in this illustration, multiple sperm release enzymes from the acrosome to digest the outer follicle cells. This cooperative tunneling targets the zona pellucida, the final glycoprotein wall before the egg's actual membrane. But this cooperative breach is dangerous. If the swarm is too successful and more than one sperm enters the egg, the resulting genetic overload instantly destroys the embryo. The egg survives by inviting a swarm to dismantle its own outer defenses, only to leave those same allies behind the exact millisecond a single cell crosses the threshold. This reaction starts in a fraction of a second. It triggers the moment the first sperm binds to a receptor and fuses its plasma membrane with the egg. This fusion activates the fast block. It forces sodium-ion channels to snap open, allowing a flood of positively charged particles to rush in. This influx causes depolarization, violently flipping the electrical charge from negative to positive. This depolarization acts like an electrical shockwave, instantly repelling other sperm that try to fuse in the immediate aftermath. It's an effective defense, but it's only a temporary patch. It buys the egg just enough time to build something permanent. As the electrical alarm sounds, the egg's internal systems respond. A signal transduction pathway commands the endoplasmic reticulum to release its internal stores. This creates a massive wave of calcium ions that physically sweeps across the entire interior of the egg. This wave triggers the cortical reaction. Thousands of tiny vesicles called cortical granules rise and fuse with the outer membrane, dumping their chemical payload into the space just outside the cell. These enzymes, known as zonal inhibiting proteins, spread through the gap. They mechanically snip off every remaining spermaceptor and generate massive osmotic pressure, flooding the space with water to physically push the other sperm away. Finally, these enzymes harden the entire outer structure, creating an impenetrable fertilization envelope. This permanent lockdown slams the door shut, ensuring the egg's survival by stopping the rest of the swarm in their tracks. The frantic energy of the swarm is over. Outside the egg, the runner-up sperm bump harmlessly against a wall they can no longer breach. Inside, the secure cell begins its transition. The nucleus of the successful sperm is absorbed deep into the egg's cytoplasm. The two sets of genetic material finally merge, forming the core that will drive the first cellular division. This process is a study in precision, balancing the destruction of its own outer walls with a hair-trigger security system that reacts in milliseconds. Conception is defined by this timing. Rather than waiting for a winner, the egg actively controls the gates, ensuring the very swarm it needed to survive is silenced the moment its work is done.